Chapter 3: The Molecules of Life. Organic compounds are carbon-based molecules. All life on our planet, as we know it, is carbon-based. Carbon is versatile because it can form up to 4 covalent bonds that can branch off in 4 different directions. The carbon atoms of organic compounds can also bond with other elements. Usually, the carbon atoms will bond with hydrogen, oxygen, and/or nitrogen. There are 4 classes of molecules that are important for life. These include carbohydrates, lipids, proteins, and nucleic acids. Very large molecules are often referred to as macromolecules. The word macro means big. Many macromolecules, such as proteins, lipids, and carbohydrates, are called polymers. Polymers are large molecules made by stringing together many smaller molecules called monomers. Monomers are linked together to form a polymer through a dehydration reaction. A dehydration reaction involves the removing of a molecule of water. A molecule of water is formed by the release of 2 hydrogen atoms and 1 oxygen atom from the reactants. A polymer grows in length when an incoming monomer and the monomer at the end of the polymer each contribute atoms to form a water molecule. The monomers replace the lost covalent bonds with a bond between one another. The breakdown of polymers occurs by a process called hydrolysis. The word hydrolysis literally translates to cut and water. The bonds between monomers are broken by adding water to them. This process is essentially the reverse of the dehydration reaction. Hydrolysis reverses the process by adding a water molecule, which breaks the bond between the 2 monomers, creating 2 smaller molecules from the larger one. Carbohydrates include monosaccharides, disaccharides, and polysaccharides. Monosaccharides are made up of 1 sugar monomer. Disaccharides are made up of 2 sugar monomers, and polysaccharides are made up of many sugar monomers. Monosaccharides are the monomers of carbohydrates. Examples include glucose and fructose. Glucose is the primary sugar type that is used by cells of the body. Fructose is known as fruit sugar. Glucose and fructose are examples of what are called isomers. Isomers are molecules that have the same molecular formula but different structures. Isomers have different properties. It is the rearrangement of functional groups that makes fructose taste much sweeter than glucose. A disaccharide is constructed from 2 monosaccharides through a dehydration reaction. Examples of disaccharides include lactose, maltose, and sucrose. Sucrose is made by 2 monomers, glucose and fructose. Lactose is made by the 2 monomers galactose and glucose. Maltose is made by combining 2 glucose monomers together. Lactose is known as milk sugar. Maltose is known as malt sugar. Maltose is used in making beer, liquor, malted milk shakes, and malted milk ball candies. Sucrose is also known as table sugar. Both monosaccharides and disaccharides are commonly referred to as simple sugars. Polysaccharides are commonly referred to as complex carbohydrates. Polysaccharides are long chains of sugars or polymers of monosaccharides. Starch is a polysaccharide found in plants. Starch consists of long strings of glucose monomers. Plant cells can withdraw glucose from starch for energy or to build materials. Glycogen is a polysaccharide used in animals to store excess fat. Glycogen is a polymer of glucose monomers, but glycogen is much more extensively branched than other types of polysaccharides. Much of your glycogen is stored in the liver and muscle cells, which break down the glycogen to release glucose when you need energy. After a day or so, unused glycogen is broken down, and its energy is then used to make fats for long-term storage. Cellulose is also a polymer of glucose, but its glucose monomers are linked together in a unique way. The bonds in cellulose cannot be broken down by any enzyme produced by animals. Grazing animals are able to derive nutrition from cellulose because of microorganisms that inhabit their digestive tracts. These microorganisms break down the cellulose in grazing animals, giving the animals the nutrition they need. The cellulose in plants that humans eat is commonly referred to as dietary fiber. Fiber clears out the digestive tract. However, it provides no nutritional benefits or calories. The health benefits of dietary fiber include lowering the risk of heart disease, diabetes, and gastrointestinal diseases. Foods rich in fiber include fruits, vegetables, whole grains, bran, and beans. Lipids are hydrophobic or water-fearing. Lipids include both fats and steroids. Triglycerides are a typical type of fat consisting of a glycerol molecule joined with 3 fatty acid tails. Triglyceride molecules are formed by dehydration reactions. Fats function to store energy in the long term. It cushions vital organs, and it insulates us to keep us warm. Saturated fatty acids contain the maximum number of hydrogen atoms, giving them a straight shape. Saturated fatty acids tend to be solid at room temperature. Examples of these are butter, lard, and shortening. Unsaturated fatty acids contain at least one double bond, causing a bend or a kink in the fatty acid. Unsaturated fatty acids are lower in energy and lower in calories. These tend to be liquid at room temperature. An example of unsaturated fatty acids are oils. Steroids are also considered lipids. All steroids have a carbon skeleton with 4 fused rings. Cholesterol is a key component of membranes that surrounds your cells. The bad type of cholesterol is LDL, or low-density lipoprotein. This is associated with higher risks of cardiovascular disease. Cholesterol is considered a base steroid from which your body produces other steroids. The hormones testosterone and estrogen, which are responsible for the development of male and female sex characteristics, are formed from cholesterol precursors. Proteins are polymers of amino acid monomers. Proteins are instrumental in almost everything that cells do. The major types of proteins include structural proteins, which provide support, storage proteins, which provides amino acids for growth, contractile proteins, which allow for movement, transport proteins, which helps to transport substances, and enzymes, which speed up chemical reactions. All proteins are made by stringing together a common set of 20 kinds of amino acids. Every amino acid consists of a central carbon atom bonded to 4 covalent partners. 3 of those attachments are common to all 20 amino acids. These are a carboxyl group, an amino group, and a hydrogen atom. The variable component of an amino acid is called the side chain, or R for radical group. Each type of amino acid has a unique side chain which gives that amino acid special chemical properties. Proteins are formed from amino acid monomers through dehydration reactions. The bond that joins adjacent amino acids is called a peptide bond. The resulting long chain of amino acids is called a polypeptide. A functional protein is one or more polypeptide chains precisely twisted, folded, and coiled into a molecule of a unique shape. For example, red blood cells of humans are normally disc-shaped. Each cell contains millions of molecules of the protein hemoglobin. Hemoglobin transports oxygen from the lungs to all of the cells of the body. In a condition called sickle cell anemia, there is a slight change in the amino acid sequence of the hemoglobin. In sickle cell disease, the inherited substitution of one amino acid, valine, in place of the amino acid glutamic acid occurs at the 6th position of the polymer. Such abnormal hemoglobin molecules tend to crystallize. This deforms some of the red blood cells into a sickle shape. Someone with the disease suffers from dangerous episodes when the angular cells clog tiny blood vessels and inhibit blood flow. Some proteins can even cause disease. These proteins are called prions. Prions can cause fatal weight loss in several animals, including deer, elk, and moose. And some cause mad cow disease. The prion converts normal proteins to become misshapen and clumped together, causing several prion diseases. Nucleic acids are macromolecules that store information and provide the instructions for building proteins. There are two types of nucleic acids: DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. DNA is the genetic material that we inherit from our parents. DNA resides in the cell as one or more very long fibers called chromosomes. A gene is a unit of inheritance encoded in a specific stretch of DNA that programs the amino acid sequence of a polypeptide. Dehydration reactions link individual nucleotide monomers together into long chains called polynucleotides. In a polynucleotide, nucleotides are joined by covalent bonds between the sugar of one nucleotide and the phosphate of the next. This results in a sugar-phosphate backbone. Each DNA nucleotide will have one of 4 bases: adenine, guanine, thymine, or cytosine. These bases hang off the backbone like appendages. The genetic code refers to the different combinations of the 4 bases in the DNA. DNA is double-stranded. There are 2 polynucleotide strands coiled around each other to form the double helix. The 2 strands must base pair together, but there are rules. The base adenine can only pair with thymine, and guanine can only pair with cytosine. If you know the sequence of bases along one DNA strand, you can then derive the sequence along the complementary strand in the double helix. The sugar in RNA is ribose rather than deoxyribose, and instead of the base thymine, RNA uses a base called uracil. Except for the presence of ribose and uracil, the RNA polynucleotide chain is identical to the DNA polynucleotide chain. RNA is usually found in living cells in a single-stranded form, whereas DNA exists as a double helix. Thank you for watching.